Hi. While collecting some water samples from a local park pond here in Tucson, I ran across a bunch of these on the same slide, and I tried unsuccessfully to ID them. Luckily, the folks over at photomicrography.net were able to identify the colony as a bacteria known as Planctomyces Bekefii. Turns out this was discovered by Nandor Gemesi in 1924. He was a teacher at the Buda St. Emmerich Gymnasium of the Cistercian order. It was located in Budapest, Hungary. He named it after the order's founder, Remigius Bekefii. The colony was originally identified as a planktonic fungus because of the rosette arrangement. It has since been discovered to be a bacteria, but not just any bacteria. Actually, this is a pretty unique one. Both gram-negative and gram-positive bacteria have cell walls composed of peptidoglycan. In gram-positive, it's a thicker wall, something like 50 nanometers, and in gram-negative, it's a thin wall, about 5 nanometers. But all bacteria have it, except for this order of bacteria. So what we have here is a gram-negative bacteria that lacks a peptidoglycan cell wall, thus making it very unique. I tried reading a few recent papers on it, and I hardly could understand a sentence. So I didn't learn too much. So here's an image that I took of this bacteria using a lights divert in phase contrast. Used a 100 power fluoride objective with a numerical aperture of 1.35 and a Leitz-Hein phase condenser. So I got a pretty nice picture of it. The white halos are artifacts of phase contrast, so they're not real. So now I've put some dimensions on the various parts of this colony. The cell bodies are spherical and about 2 microns in diameter. They have a non-cellular stalk attached to each one, and it's about 6 microns long in this colony and about 300 nanometers in diameter. The bacteria reproduce by budding, and it looks like I've caught 2 daughter cells budding off 2 of the main cell bodies. And apparently when the daughters mature enough, they leave the colony as what's called a swarmer with a single flagellum. And that flagellum, I think, is only visible with an electron microscope. Here's another 2 colonies imaged in the same way that the first one was. The stalks are a bit longer, about 8 microns. Otherwise, same overall dimensions. Phase contrast, again, plays some tricks. The out-of-focus bodies are white, whereas the in-focus ones are a darker color. And again, I think I caught a budding daughter cell on the left. Anyway, this was interesting. It got me learning at least a tiny bit about bacteria and the uniqueness of this one in particular. So I thought more into putting it out there as a video. Hopefully you found it interesting like I have, and I didn't mispronounce too many things along the way. Anyway, thanks for watching and take care.